does our body decide when to store sugar versus spend sugar to form more energy? As we've learned over the last few lectures, glycogen acts as our primary storage of carbohydrates, or our glucose, within the body. We can take our glucose molecules and, of course, link them together to form glycogen using the pathway shown here. Right? Our glucose is converted to glucose 6-phosphate. We move that over to the first position to make glucose 1-phosphate, then produce our UDP glucose before finally linking that onto our growing glycogen chain. On the reverse, if we need extra sugar for our body to use for energy, we can break off individual glucose residues with our glycogen phosphorylase. These come off as glucose 1-phosphate, which then is transformed into glucose 6-phosphate and glucose. If we want to control when we are storing sugar versus releasing sugar from glycogen, we're going to, of course, do this at our key irreversible points in our pathway. For glycogen, this is going to be our glycogen synthase, right, which drives the actual addition of glucose onto our glycogen polymer, and the glycogen phosphorylase, which, of course, breaks off those individual glucose residues through that phosphorylase reaction. So our glycogen phosphorylase is going to be our key enzyme controlling glycogen breakdown. As with many of our glycolysis and gluconeogenic enzymes, it's going to be regulated through phosphorylation. So we have a phosphorylase B kinase shown over here on the right. This will phosphorylate our phosphorylase to create an active enzyme. So this would then lead to our glycogen breakdown, releasing extra sugar to the body. And this is going to be triggered through glucagon. So glucagon is going to activate our phosphorylase B kinase. Epinephrine will also activate it, although this will occur in the muscle, whereas glucagon occurs in the liver. On the other hand, of course, we have a protein phosphatase here that's going to be inhibiting our phosphorylase by dephosphorylating it, which this, of course, will be activated by insulin, and we'll look more at this in detail later. As usual, insulin is triggering the dephosphorylation. Glucagon is triggering the phosphorylation. Glucagon, of course, is telling us that we are low on sugar and we need to then be releasing more sugar from our glycogen in order to feed our body. Now, the stimulation of our glycogen phosphorylase kinase actually occurs through a signaling cascade. So our epinephrine and our glucagon both bind to receptors in the cell membrane. This triggers our G protein, which then leads to the production of cyclic AMP. So it's just a few molecules binding at the membrane. We can produce 10 times more of our cyclic AMP. Cyclic AMP then, of course, activates our protein kinase A, which is also our cyclic AMP dependent kinase. And this is what ultimately will phosphorylate and activate our phosphorylase B kinase in order to ultimately trigger our glycogen breakdown and glucose release. So this sort of signaling cascade is a great way to amplify a signal that our cells receive. Right? So notice how we start with just a few molecules binding at the membrane, triggering cyclic AMP release. That triggers our protein kinase become active, which then, of course, activates our phosphorylase, which can end up cutting off thousands of glucose molecules from our glycogen in order to release that to our body. So this sort of cascade is one of the ways that we can take just a few hormone molecules and then create a very large response from those molecules. Now, of course, in addition to that hormonal regulation, we're also going to have some local regulation here as well. So notice we have glucose 6-phosphate, ATP, and AMP all involved here. Glucose 6-phosphate is going to act as our kind of substrate level regulator. It inhibits our phosphorylase, right? We already have excess glucose 6-phosphate. There's no need to be breaking down glycogen which is ultimately going to produce more glucose 6-phosphate. So this is essentially a negative feedback mechanism where our product of glycogen breakdown is inhibiting our enzyme that's going to be ultimately producing that glucose 6-phosphate. Of course, our ATP and AMP here are acting as our local energy-based regulation. So, of course, if we have lots of energy available, lots of ATP, then we don't need to break down glycogen, right? Glycogen is where we store our energy, so we'd rather be making more glycogen with that excess energy than breaking it down. So ATP will inhibit our phosphorylase, whereas AMP will activate it. So in cases of low energy, we want to increase our glycogen breakdown. And if we have lots of energy, then we need to, of course, inhibit our glycogen breakdown. Now, beyond our local regulation here, of course, we also have that hormonal regulation. So here's that phosphorylase kinase that we showed previously. Again, we have glucagon triggering this through that long signaling cascade, ultimately leading to our protein kinase A, activating our phosphorylase kinase and activating glycogen breakdown. So again, this would be a glycogen breakdown signal. Now, as I mentioned, insulin is going to be our key regulator on the other side. So insulin is going to activate a protein phosphatase, which is dephosphorylating actually both our phosphorylase itself, but also our phosphorylase kinase. So you can notice that's happening up here at the same time that we're deactivating our phosphorylase B here, our glycogen phosphorylase, in order to slow down that glycogen breakdown. So insulin not only shuts down the enzyme itself that's actually breaking down our glycogen, but it also is shutting down the enzyme up here that is working to activate our glycogen breakdown. So it kind of has a dual role there in terms of its activity. Now, of course, on the other hand, our glucagon, again, will also want to slow down this effect of insulin when we want to increase glycogen breakdown. So it's actually a second mechanism here for glucagon, where it uses our protein kinase A to activate this protein phosphatase inhibitor down here. So we phosphorylate it and activate it, which is then used to inhibit our protein phosphatases in order to stop the action of insulin. Right? So when we get that glucagon signal, we essentially first turn off the insulin signal by deactivating all of our protein phosphatases, and then we use our phosphorylase kinase to activate and turn on our glycogen breakdown. So essentially, our glucagon is using this phosphoprotein phosphatase inhibitor, that's right here, in order to inhibit the insulin's signal before, of course, increasing glycogen breakdown itself. So the sort of multi-layered hormonal regulation makes sure that, again, we're always controlling exactly when we're breaking down glycogen versus, of course, synthesizing glycogen. Insulin immediately will try to stop our glycogen breakdown, as, of course, that would be indicating high levels of glucose, whereas our glucagon immediately wants to halt that insulin-based trigger and then increase our glycogen breakdown to release more sugar. Now, we also have an interesting mechanism here that glucose can be used in order to help with our regulation here as well. So within our glycogen phosphorylase, so of course, again, it's activated when we have these phosphate groups added on to our phosphorylase. But these phosphate groups actually get kind of hidden inside of our phosphorylase when it's active. When we bind glucose in these two allosteric sites, it actually causes a change in conformation in order to present our phosphate groups where they can then be more easily removed by our protein phosphatase. So glucose binds, triggers these to pop out, and then insulin can trigger that dephosphorylation with our protein phosphatase. So of course, again, if we have excess glucose levels, we want to be stopping our glycogen breakdown so that we're not wasting our glycogen in order to produce sugar that we already have. So it's kind of another negative feedback mechanism similar to the glucose 6-phosphate-based mechanism. So now let's look at the other half of our regulation, our glycogen synthesis. So of course, glycogen breakdown, we want to trigger with glucagon, and then our insulin is going to be inhibiting that at the same time. Glycogen synthesis is going to be controlled by insulin, right? So insulin will trigger our glycogen synthesis, and of course, glucagon will be trying to inhibit it at the same time. So our insulin signaling pathway is going to increase glucose import into muscle, stimulate the activity of our muscle hexokinase, and then activate our glycogen synthase so that we get that excess blood sugar flowing into our cells and then into our glycogen storage. Of course, that increased hexokinase activity at the same time as activating our glycogen synthase is critical in order to ensure that we have sufficient glucose available in our cells, glucose 6-phosphate, in order to actually make glycogen. So this figure kind of illustrates that here, where our insulin is actually going to be acting in multiple areas at the same time. It increases our glucose transporters here in order to bring more of that glucose into our cells. Also, we increase our hexokinase activity through some of the mechanisms that we've discussed previously. And then we're going to be specifically looking at our glycogen synthase here. So here's our glycogen synthase, and it's going to be activated again when it's dephosphorylated. So this is our dephosphorylated form over here on the left. And this is also the activated form. Again, as usual, insulin tends to dephosphorylate things in order to activate them. Glucagon more often activates things. So notice, of course, again, we have our protein phosphatase down here, which is triggered by insulin in order to dephosphorylate our glycogen synthase. Of course, glucagon and epinephrine are inhibiting this through that protein phosphatase inhibitor that we saw previously. If we want to deactivate our glycogen synthase, we're going to use two different kinases. So we have this casein kinase, which does a priming phosphorylation. Once it adds a single phosphate, then our glycogen synthase kinase comes along and actually adds on 3 phosphates. So of course you can see those 3 phosphates represented here on the right, and that will then shut down our glycogen synthase to stop our glycogen synthesis. Just like glucagon inhibited our insulin that was activating our glycogen synthase, our insulin is also going to block the glycogen synthase kinase that's trying to deactivate that glycogen synthase. So again, they're both working together. Insulin is working to increase glycogen synthesis. And our glucagon is looking to inhibit our glycogen synthesis. So at this point now, we have a good idea of our overall regulation of carbohydrate metabolism. right? So we have our two key hormones, insulin and glucagon. They're both working together in order to make sure that we are maintaining our blood sugar concentrations without letting it get too high or too low, leading to, of course, starvation or excess sugar within the bloodstream. So if we have high blood sugar, then of course our signal is going to be triggered through insulin. So insulin is our main signal of those high blood sugar levels. We're also, of course, going to be using transporters to increase the amount of glucose inside of our cells, right? So we increase our glucose concentration. That leads to more glycolysis and glycogen synthesis through mechanisms like, of course, hexokinase 4 is activated by increased glucose concentrations. With our glycogen synthesis, we have the inhibition of our glycogen phosphorylase that occurs as that glucose concentration increases. We also increase the activity of our glycogen synthesis because, of course, we have more of our substrate available there. Insulin, of course, works through several different mechanisms. So it does have some kinases that will stimulate initially, as well as increasing the production of some of our key glycolytic enzymes. So notice our synthesis of hexokinases, PFK1, pyruvate kinase are all increased, as we saw earlier in the presence of insulin. Ultimately, our kinases will trigger our key phosphatase here, as well as inhibit our glycogen synthase kinase. And that phosphatase, of course, is what will inhibit our phosphorylase kinase to block glycogen breakdown and activate our glycogen synthase in order to increase glycogen synthesis. On the reverse side, low blood sugar is detected by glucagon. Glucagon, of course, works through our cyclic AMP, which then triggers pKa, our key cyclic AMP dependent protein kinase. As we've seen, pKa regulates a bunch of different enzymes through phosphorylation, right? We activate our phosphorylase kinase, we inhibit our glycogen synthase, and as well as our protein phosphatase. We deactivate our pyruvate kinase and our PFK2 and FBPase2, of course. This is through that phosphorylation, which activates the bisphosphatase activity. Both of these lead to inhibition of glycolysis, as we want to slow down the expenditure of glucose in order to prevent our blood sugar from dropping too low. Of course, our phosphorylation of the phosphorylase kinase then activates glycogen breakdown, which will be releasing more sugar to our bloodstream in order to, again, counteract that low blood sugar. So this shows just kind of a nice overall summary of what our two key signals here are doing between insulin, which is driving a decrease in blood sugar as we're trying to use up extra glucose, versus our glucagon, which is trying to drive an increase in blood sugar as we're trying to raise up that low blood sugar. Now, between the liver and our muscles, there will be a slight difference in terms of how we respond for our carbohydrate metabolism. So the liver, of course, again, is going to be sensitive to glucagon, whereas epinephrine will be more directly responsible in our muscles. The muscle, of course, is primarily just concerned with itself, right? If it needs more sugar, it's mostly just going to be breaking that down and immediately using that for energy, right? So epinephrine is going to increase our glycogen breakdown, or our glycogenolysis, you could also call it. And it's also going to be increasing glycolysis. On the other hand, of course, gluconeogenesis itself is not going to be affected here. In the liver, our epinephrine is actually going to trigger gluconeogenesis in order to release more sugar to the rest of the body. So there's a little bit of a difference there in terms of the liver versus our muscle, where in the liver we're concerned about shipping out glucose to the rest of the body, whereas in the muscle We're really just concerned about using our own glucose to maintain our energy sources. So now that we have a firm understanding of our carbohydrate metabolism and how we want to regulate that using insulin and glucagon, we're going to take a deeper look at the aerobic metabolism of pyruvate and how that pyruvate feeds into the citric acid cycle. Ultimately, of course, this will also lead into our other key metabolic pathways, right? So our fatty acids and our amino acids will also feed into that same citric acid cycle. So those will all kind of share that same component of our aerobic metabolic pathway.